Welcome to The Explainer. Today, we're going to peek under the hood of one of the most important processes in all of science, the clinical trial. We're going to unpack the amazing statistical machinery that lets researchers say with real confidence, hey, this thing actually works.
So let's just jump right in with a question that, was on pretty much everyone's mind a few years ago. The whole world was holding its breath waiting for a vaccine, and the size of those trials, they were just massive. So why on earth did they need a group of volunteers the size of a small city?
Well, here's the thing. That huge number wasn't just a number they pulled out of a hat. Nope. It was the result of a super careful, deliberate calculation. The whole point was to build a study so powerful it could clearly tell the difference between a real effect and just the random noise of life. And today, we're going to walk through exactly how they did it.
Okay, first things first. We have to understand the fundamental problem that every single scientist faces, no matter what they're studying.
Right. So think about it like this. Let's say you're testing a new pill for headaches. Someone takes it, and poof, their headache's gone. That's great. But wait a second. Would it have gone away on its own anyway? Maybe they just needed a glass of water. How can you be 100% sure it was the pill? That right there is the core challenge, separating a real effect from just pure random chance.
So to solve this puzzle, scientists basically make a formal bet. It's a powerful statistical framework, and it's called hypothesis testing.
The starting line for this bet is something called the null hypothesis. You can think of this as the ultimate skeptical position. It basically assumes the new treatment, the new drug, the new vaccine does absolutely nothing. Zero. Zilch. It's kind of like the innocent until proven guilty rule in a courtroom. The default assumption is that there's nothing going on.
And then on the other side of the ring, you have the alternative hypothesis. This is what the researcher is actually trying to prove. This is their big idea, their claim that, yes, my treatment really does have a measurable effect. The entire experiment is designed to see if they can gather enough evidence to knock out that null hypothesis. So bringing this back to the COVID vaccine trials, the scientific bet was set up just like this. The null hypothesis was, this vaccine is no better than a saltwater shot. And the alternative was, no, this vaccine actually works and lowers infection rates. The whole goal of that gigantic trial was to collect enough powerful data to confidently say, we can throw out the null hypothesis.
Now, making this bet is a really high-stakes game. Why? Because there are two major ways you can get the answer wrong, and honestly, both of them have some pretty serious consequences.
This chart lays it all out. On one hand, you could make a type 1 error. That's a false positive. It's a false alarm. It's when you conclude the treatment works, but in reality, it doesn't. You just got fooled by random chance. And on the other hand, you have a type 2 error. This is a false negative. This is a huge missed opportunity. This is when the treatment really does work, but your study just wasn't powerful enough to see it.
And this is exactly why getting that number of participants right is so critical. It's an ethical tightrope. A trial that's too small risks missing a life-saving drug, which is a tragedy. But a trial that's too large is also a problem because you're exposing more people than you need to an experimental treatment that might have side effects. It is a very fine line to walk.
Which brings us right back where we started. How in the world do scientists find that perfect Goldilocks number of people? Not too few, not too many, to make sure they minimize the risk of making one of these huge errors.
Well, it's not magic, it's a recipe. A precise recipe with 4 key ingredients. First, you have your significance level. That's the risk you're willing to take of a false alarm, usually about 5%. Then there's power. That's your confidence that you'll actually spot a real effect if it's there. You want to aim for 80 or 90%. Third, effect size. This is about how big of a change you're looking for. A really subtle effect needs a way bigger sample than a huge dramatic one. And finally, you've got variability. Basically, how noisy is your data? The more scattered the results are naturally, the more people you need to see a clear signal through the noise.
And this is the cool part. Here's how those ingredients were used in the real world. Just look. Pfizer, Moderna, AstraZeneca, they all set very similar goals. They all wanted around 90% power to be able to detect a vaccine that was at least 60% effective. And because they knew the infection rate in the general population was going to be pretty low, they knew they would need a ton of people to see a difference that was statistically solid.
And that, right there, is how Pfizer, after plugging all those ingredients into their statistical formulas, landed on their target. 21,999 people getting the vaccine and 21,999 getting the placebo, a grand total of almost 44,000 people. It wasn't a guess. It was the exact number they calculated they'd need to get a result they could trust.
Okay, so the trial is done. The data from all those thousands of people comes pouring in. Now what? How did the scientists make the final call? How did they deliver the verdict?
They use a tool called a test statistic, like the z-statistic you see here. Now, you might have heard of a z-score, which tells you how one single person compares to the average. A z-statistic is kind of like that, but on steroids. It looks at the entire trial, the average result from all 44,000 people, and it asks one simple, powerful question. Is this result so weird, so far from what we'd expect by dumb luck, that something real has to be causing it?
And that statistic is the star of a super clear four-step process. First, you state your hypotheses, we've talked about that. Then you calculate your test statistic from all your data. Third, you compare that number to a critical value you decided on beforehand to see if your result is extreme enough. And finally, you make the call. You either have enough evidence to reject the null hypothesis and celebrate, or you don't. It's as simple as that.
So this whole thing, from making the initial bet, to calculating the exact number of people, to running the final numbers, that whole rigorous process is what provides objective mathematical proof. It's how we take a promising idea and turn it into a scientifically validated conclusion that we can all rely on.
And that really brings us to the takeaway here. The next time you see a headline about some amazing new scientific breakthrough, just think about this whole process. Think about the thousands of volunteers, the incredibly careful calculations, and this whole statistical framework that's designed to protect all of us from being fooled by random chance. Knowing what goes on behind the curtain, does it change how you see that news?